Good morning, all of you. Myself, Payal Prajapati, and I am working as a teaching assistant in HVHP Institute of Postgraduate Studies and Research Kadi. Today, I am going to introduce the microbial growth curve. So, let's move ahead. Bacterial growth. All living organisms have the fundamental ability to the growth. Growth, that means the formation of new cell material and increasing in the number of cells, not only an increasing in the size due to cell division. Growth is defined as the balanced increasing in all cellular constituents followed by the increasing in cell number. Simple increasing in cell mass may not represent the growth. Increasing in the cell mass may due to the increase the reserve material in the cell such as the glycogens and polybihydroxybutyrate or water content. With the balanced increase in the cell mass, all cellular constituents such as the cellular nucleic acid, proteins and other cellular molecules increase the proportionally. You can see in the slide that is the bacterial growth or the bacterial plate. Growth is generally used to refer the acquisitions of the biomass leading of the cell divisions or the reproductions. Growth requirements, its need to the nutrient for their growth. Organism use a variety of nutrients for their energy needs and build organic molecules and cellular structure. Most common nutrient, those containing necessary elements such as the carbon, oxygen, nitrogen, and hydrogen. Microbes obtain nutrients from variety sources. When bacteria are inoculated in the fresh medium and allow their growth, they show characteristics for the growth pattern. This is known as normal growth curve of the bacteria. This pattern is observed in the batch or closed culture system. Batch culture is the one in the which bacteria once inoculated in the medium are allowed to complete their growth without further adding any nutrient in the medium. It is also closed culture because once the organism is inoculated in the culture medium, they are allowed to complete their growth in the same medium. It's called as the batch culture, either you can say the closed culture medium. During the incubation's nutrient concentration decline and the concentration of the waste increase. The growth of microorganism can be plotted as the logarithm of the number of the viable cell versus the incubation time. In the resulting curve has a 4 distinct phase. The first one, that is the lag phase. You can also say that the adaptation as well as the initial phase. Second one, that is the log phase. You also known as that is logarithmic phase. Either you can say the exponential phase. In the third one phase, that is the stationary phase. You can say the stagnant phase. And the fourth one, that is the death phase. You can say also it decline phase. The occurrence of the different growth phase is due to the constantly changing environment in the medium due to the bacterial activity. You can see in the graph time versus number of the bacteria. You also say that by this 4 distinct phases are there: lag phase, log phase, stationary phase, and the decline phase. So let's study one by one phase. When bacteria are inoculated in the fresh medium, initially for some time, there is no visible increase in the cell number and cell mass. This is referred to as lag phase of the growth. This attributes to various reasons. First one, if the inoculum is obtained from old culture or culture from death phase, it may carry with the toxic metabolites which may continue to inhibit the growth. Such cell may be also metabolically weak, having a very few number of ribosomes, RNA, and the protein. Much often, the nutrient environment in the fresh medium may be quite different than the one that might be in the old or previous culture. Hence, microorganisms may require some time to adapt the new environment. If the inoculation is obtained from spore-forming bacteria, and consist mainly of spores. They must be germinated and get converted to the vegetative cells before starting the growth. This time for spore germinations may also count for the leg phase of the growth. So this is all about the leg phase. Certain changes in the during the leg phase in the bacterial cell. So there is no visible increasing in the cell number during the lag phase. This is because of metabolic and physiological change do occur in the bacteria to prepare them to undergo rapid growth. Hence, we cannot consider the lag phase as ideal phase. During this phase, the number of ribosome, amount of RNA, DNA, and protein increase in the cell. This shows the physiological activations of the weight cells and the overall rate of the metabolic activity and respiration is increasing. The cell size is also increasing, but not viable increasing in the cell number. Factor affecting the length of leg phase. Let's see some factor affecting of the leg phase. First one, that is the type of inoculation. Inoculum. If the inoculum is obtained from previous culture occurring in the log phase, it will show a shorter leg phase. This is because cells from the log are metabolically more active than those occurring in the death phase. Similarly, inoculum containing a large number of spores will show the longer leg phase. Second one, that is the medium compositions. If the compositions of fresh medium is similar to that previous one, or if it is more rich, a smaller large leg phase in the optics. Third factor, that is the size of inoculum. If the inoculum is large, the culture shows shorter leg phase and vice versa. Exponential phase, it is also known as the log phase and second phase of the bacterial growth curve. By the end of the leg phase, most organisms in the culture are active and large enough to divide. Hence, they start dividing, causing logarithmic increase in the cell number. Since the environmental and the nutrient conditions are most favorable, this bacteria will show maximum rate for the growth and maximum generation time. That's why we call it as the exponential phase. Usually, the medium contain nutrient in excess. Therefore, even though nutrients are being constantly depleted from the medium due to their utilization during growth initially, the change in their concentrations has very little effect on the growth. Hence, the bacteria continue to grow in the logarithm for the many generations. However, during the growth, the chemical environment of the medium undergoes a constant change, gradually become unfavorable for the growth. This results into gradual decline in the growth rate. Hence, it increases the growth slowdown, allowing it other seasonally phase. The development of the unfavorable chemicals and physical environment of the medium occurs due to the following reason. Let's see. During growth, bacteria consume nutrient as well as the dissolved oxygen. Therefore, gradually their supplies become limited and unfavorable for the growth. Further, as growth increasing, the need for the nutrient increasing more and more. Hence, these depletions become more significant during the latter part of the log phase. Increasing in the cell mass due to the growth cause increasing in the viscosity of the medium significantly. This also provides the unfavorable condition to the microorganism for the uptake of the nutrient as well as oxygen by the organisms. Even enough space to also not available for the organism to have a free movement in the medium due to their increase in the cell density. Due to the respiratory metabolic activities of the organism during growth, drop of pH of the medium is normally observed. This also contributes to the development of the unfavorable condition for the organism. The metabolic activities of the organism result into the excretions of the various waste metabolic by the organism. These metabolites may have toxic effect on the microorganism and they have unfavorable effect on the growth, tend to retard growth. During the exponential or log phase, microorganisms are growing and dividing at the maximum rate possible given their genetic potentials, the nature of the medium, and the conditions under which they are growing. This rate of growth constant during the exponential phase, that is, the microorganisms are dividing doubly in the number at the regular intervals. Because each individual divides at the slightly different moment, the growth curve rises smoothly rather than in discrete jump. The population is most uniform in the term of chemical and physiological property during this phase. Therefore, exponential phase culture are usually used in the biochemical and several physiological studies. Exponential growth is balanced growth. That's a cellular constituent are manufactured and the constant rate relative to each other. If nutrient level or other environmental conditions change, unbalanced growth result. This is growth during which the rates of synthesis of cell component vary relative to one another until a new balanced state is reached. Unbalanced growth is readily observed in the two types experiment. First one, that is the sit up, and the second one, that is the sit down. In the sit up, culture is transferred from nutritionally poor medium to a rich one. In the sit down, where culture is transferred from a rich medium to a poor one. So, in the sit up, there is a lag while the cell first construct a new ribosome to enhance their capacity for the protein synthesis. This is followed by the increasing in the protein and DNA synthesis. Finally, the accepted rise in the reproductive rates take place. In the sit down experiment, there is a lag phase in the growth because of the cell need to time to make an enzyme required for the biosynthesis of unavailable agent. Consequently, cell divisions and DNA replication is continued after the sit down, but net protein and RNA synthesis is slow. The cell becomes smaller and reorganize themselves metabolically until they are able to grow again. Then balanced growth is resumed and the culture enters the exponential phase. This shift up and shift down experiment demonstrate that microbial growth is under precise, coordinated control and respond quickly to change in the environmental conditions. When the microbial growth is limited by the low concentrations of the required nutrient, the final net growth or yield of cells increase with the initial amount of the limiting nutrient presence. The rate of growth also increases with the nutrient concentrations, but in a hyperphobic manner, much like that seen with the many enzymes. At sufficiently high nutrient level, the transport systems are saturated and growth rate does not rise further with increasing nutrient concentrations. So you can see in the figure nutrient concentrations and their growth in A1 graph plot, that is nutrient concentration versus total number of cells, and B1 nutrient concentrations versus growth rate. In A, the effect of changes in the limiting nutrient concentrations on the total microbial yield at the sufficient high concentration and total growth will be plated. And second one, the effect of in the growth rate. You can see in the flow charts. Third one, that is the stationary phase. You can also say that is the stagnant phase. Because this is a closed system, eventually population growth cause and the growth curve becomes horizontal. The stationary phase usually is attained by bacteria, a population level of around 10⁹ cell per ml. Other microorganisms normally do not reach such a high population density, Protist culture often have maximum culture about 10 raised to 6 cell per mm. So finally, population size depend on the nutrient availability and the factors as well as type of microorganism being cultured. In the stationary phase, the total number of viable microorganisms remain constant. This may result from balance between the cell division and cell death, or the population may simply cause to divide but remain metabolically active. Microbial populations enter the stationary phase for the several reasons. One reason is nutrient limitations. If an essential nutrient is severely depleted, population growth will slow. Aerobic microorganisms often are limited by oxygen availability. Oxygen is not very soluble and may be depleted of the quickly that only the surface of the culture will have an O2 concentration adequate for the growth. The cells band, the surface will not be able to grow unless the culture is shaken or aerated is in another way. Population growth also may cause due to the accumulations of the toxic waste product, and you can say toxic the metabolic product. This factor seems to limit the growth of many anaerobic cultures. For example, streptococci can produce so much of the lactic acid, other organic acid from sugar fermentation. That's the medium becomes acidic and the growth is limited, inhibited. The growth may be caused when a critical population level is reached. Thus, and turns into the stationary phase may be result from a several factor operating is concerned. The bacteria is batch culture may enter the stationary phase is response to these starvations. This probably often occurs if the nature become many environments have low nutrient levels. Prokaryotes have evolved a number of strategies to survive starvation. Many do not respond with obviously morphological changes such as endosperm formations, but only decrease somewhat in overall size, often accompanied by the protoplast shrinkage and the nucleoid condensation. The more important changes are in gene expressions and physiology. Starving bacteria frequently produce a variety of starvation proteins which make cell much more resistant to damage in the variety of ways. They increasing peptidoglycan cross-linkage and the cell strength also. The DPase protein, that means the DNA binding protein from starved cells. It also protects the DNA of cells. Chaperone proteins prevent the protein denaturation and renature the damaged proteins. As a result of these many other mechanisms, the starved cell become harder to the kill or more resistant to the starvation itself. Damaging temperature changes, oxidative and osmotic changes, and toxic chemicals such as the chlorine. These changes are so effective that some bacteria can survive in starvation for years. There is even evidence that Salmonella enterica and Servo's typhimurium and some other bacterial pathogens are become more virulent when it in the starvation phase. Senescence and death. For many years, the decline is viable cell following stationary cell was described simply as in the death phase. It was assumed that detrimental environment change in the nutrient deprivative and build up the toxic waste cause irreparable harm resulting in the loss of viability. That is, even when bacterial cell when transferred to the fresh medium, no cellular growth has observed because of loss of the viability was often not accompanied by the loss of total number of cells. It was assumed that cell died but did not lies. This view of currently under debate. There are two alternative hypotheses are that some microbiologists believe starving cells that show on the exponential decline in the density have not irreversibly lost their ability and reproduce. Rather, they suggest that microbes are temporarily unable to grow at at least under the laboratory conditions used. This phenomena in the which of the cells are called viable but not culturable. It's expand as a VB and C, that is viable but not culturable, is thought to be the result of the genetic response triggered in the starving stationary phase cells. Just as some bacteria form spore as a survival mechanism, it is argued that others are able to become dormant without changes in the morphology. Once the appropriate conditions are available for the VBNC microbes resume their growth. VBNC microbes could pose a public health threat as many assays that test for the food and drinking water safety are culture based. The second alternative to the simple date phase is the program cell date. In the construct to the VBNC hypothesis, whereby cells are genetically programmed to survive, program cell death predict that fractions of the microbial population is genetically programmed and committed suicide. In this case, non-culturable cells are dead and the nutrient that they lack enable the survival individual growth. Those cells is populations that die not or initiate suicide. The dying cells are thus, that is to say, they sacrifice themselves for the benefit of the larger populations. You can see in the figure over here how it lost their viability. You can see in the A, B and C. In A, it has a long been assumed that the cell leaves stationary phase due to the starvations or toxic waste accumulations. The exponential decline in the culturability is due to the cellular death. In the B, some believe that the fractions of microbial population dies due to the activations of program cell death gene. The nutrients that are released by the dying cell support the growth of other cells. In the C, the viable cell but not culturable cell. Hypothesis posed that when the cells are starved, they become temporarily non-culturable under the laboratory conditions. When exposed to appropriate conditions, some cells will regain the capacity to be reproduced. Phase of prolonged decline. It is long-term growth experiment reveal that and the exponential decline in the viability is sometimes replaced by the gradual declines in the number of culturable cells. This decline can last a month to years. During this time of the bacteria population continually evolves so that actively reproducing cells are those best able to use nutrient released by the dying, retain and best able to tolerate the accumulated toxins. The dynamic process is marked by senescence with the genetically distinct variant. Thus, the neutral selections can be within a single culture vessels. You can see the prolonged decline phase time versus the cell number. Instead of the distinct phase, successive waves genetically distinct subpopulations of the microbes better able to use the released nutrient and accumulated toxic for their survival. Each successive solid or dashed curve represented the growth of all new subpopulation. So this is all about the bacterial growth curve. I hope you liked it. Thank you for watching me.